Welcome to Spotlights. So today we'll be talking to Chee Kiang, or Ethan, who you've already seen in previous episodes. Hi, Ethan. You'll be telling us something about your exciting new preprint that's just come out. Could you maybe first introduce yourself?
I'm currently a postdoc in Oded Rechavi's lab in Israel. I'm coming up to third year of my postdoc in March. Here, I'm interested in how small RNA regulating gene expressions and executing some of the important functions in this little animal that we study, the nematode C. elegans, and also how the small RNA enable epigenetic inheritance as well.
Excellent. So what's the specific topic of your preprint then?
Yeah, in this preprint that we just put out, I was interested in how this really rapidly evolving gene family called Pulse, and specifically one member Pulse22, how it regulates innate immunity against many pathogens like viruses and intracellular fungal, but also how Pulse22 influence the functionality of RNAi.
So actually, I was wondering, could you tell us something about why this work is important?
Yeah. So RNAi is deeply conserved processes across many organisms, from plant to animals. And in C. elegans, a organism that we study has especially elaborate RNA machinery, which makes it easier or very interesting to study, which recently got a Nobel Prize for it. And so this work was important for me, and I think it's important for the field that we study how the functionality of RNAi can be quickly changed in response to genetic perturbations or in response to infections by intracellular pathogens. So, for example, a mutation in Pulse22 causes huge upregulations of some of the argonautes, which are the main effectors of RNAi, and that leads to a better immune defense against these different pathogens and also intracellular parasite as well, genomic parasite.
Can you maybe tell us what the main challenge was during this work?
The main challenge was to convince people about studying Pulse22, which is, as I mentioned earlier, it was rapidly evolving, and Pulse22 specifically, it's not found outside of C. elegans. So it's a quirky worm thing that not a lot of people care about, and it was initially hard to convince other people why studying Pulse22 is interesting. But to me, the fact that it is rapidly evolving and it's not conserved makes it more interesting, not less.
Is there something specific that makes you especially proud to have this out?
I was proud that I made the right judgment call of pursuing Pulse22, the gene that I just talked about, and it's one of the many Pulse gene and in this family that does things that we haven't yet understood. And we did discover some really interesting way RNA can be regulated, potentially beyond functions of this Pulse gene. So one of the things that we found was canonically, RNAi has thought to be functioning in gene silencing. So when the small RNA guide these argonautes bind to its target, and that leads to target degradations. That is the canonical textbook picture of RNAi. But what we found was, yes, in wild-type background, that is true. So when you remove RNAi factors, you see upregulations of immune genes. So these RNAi pathways normally target immune genes and downregulate them. But in the absence of Pulse22, you get downregulations of these immune genes when the RNAi factors are absent. So what is implying is that in the absence of POS22, these RNAi pathways somehow upregulate the immune gene expression instead of downregulating it. So that kind of going against the canonical thoughts that RNA is solely doing gene silencing. So that itself is an interesting biological phenomenon, and POS22 seems to like a switch to these different functionality of RNAi, whether it's gene silencing or gene promoting. And I think the implication of this could be beyond POS22 too, and that's what I intend to study further. Maybe there are different stresses. So we in here we focus a lot of these mutation in POS22 genes, and we know that POS22 is very important to normally repress immune system, and it has a role in immunity and dealing with different pathogenic stress. So maybe there are different stresses that could also influence this kind of switches in RNAi machinery, which I intend to study further.
So what then would be your main takeaway from your preprint? So if there's one thing that people should remember, what is it?
That corty worm thing can be very interesting.
And more specifically about, the process you uncovered?
Yeah. So the take home is that there's a rapidly evolving genetic switch that leads to different functional outcome of RNAi. And that could also, I have some indications that these processes could also affect how small RNA and epigenetic memory can be passed on from one generation to another, which is another big conversation to have, how in C. elegans small RNA can be passed on for many generations.
That's awesome. Well, thanks, Ethan. Thanks very much for sharing. If you want to learn more about his work, please check out the preprint. It's now available on bioRxiv. There will also be a link in the description. Thank you very much for listening, and see you next time.